Okay, continuing with chapter 29 on fungus. So in the last lecture, we wrapped up here, talked about how those mycorrhizal fungi can benefit some plants, and without them, we see significantly stunted growth in others. So another type of fungi that really substantially benefits the ecosystem are these saporific fungi, and what they do is they really substantially accelerate the carbon cycles. So, thinking back to maybe from Bio 1, we learned about the carbon cycle, if you remember, right? You got CO2 in the atmosphere, and plants take in CO2, and they can turn it into glucose that can then be used by the plants, then eaten by animals and things like that. Essentially, these saporitic fungi, saporitic fungi will um, speed up this process quite substantially. So before there were these fungi, specifically during the Carboniferous period, where we got a whole lot of coal, that's where all that coal came from, all that excess carbon. Today, we see these really speeding up, like I said, the terrestrial carbon cycle. All right. Um, what fungi do that's really unique, and it's kind of discussed here, is that these fungi, they can digest cellulose, lignin, things that other organisms cannot, and therefore recycle the carbon in those molecules. So within cellulose and lignin, so these really tough polysaccharides that we can't digest them, like you can't eat grass and digest that, whereas fungi can actually take dead organisms, like dead grass, and turn it back into carbon that can then be used. All right. So without that saporitic fungi, the carbon would just sit there in dead wood and things like that, and would never, recycle back into the environment, and we'd see a much less productive environment. So I hope that makes sense as to why these are really important to the carbon cycle. They speed it up. They can take carbon out of dead organisms that we can't, that other organisms cannot. So it's really extraordinary that they can do that. All right, so morphological traits, very simple fungi in terms of their morphology. So two growth forms of fungi exist. There's the single-celled forms like yeast, okay? And then there's multicellular filamentous mycelia, which we'll see, okay, singular mycelium. Some species have adapted both forms of that at some point in their life cycle, which we'll see. Okay, so here's the single-celled fungi here in the electron micrograph, the yeasts, right? And more commonly, if we look at multicellular fungi and we look inside their bodies, we see that they have this web-like structure called mycelia. And if we look closer at this mycelia, we'll see it's made up of branches called hyphae. So if we examine the mycelia, they're constantly growing in the direction where food sources are. So they're dynamic. Okay, they're going to die back in an area where food is running out. So they kind of put their feelers out and can determine where the food is. The body shape of fungus can also change continuously throughout its life. So these are, again, very dynamic organisms. And when we look at these mycelia, we see that they, are very long-lived, very large, and very beneficial to the fungi. So if we look at the nature of hyphae, okay, so these are the long, narrow, frequently branching filaments that actually make up the mycelium. So there's the hyphae uh, in there, okay? And if we look kind of inside of these hyphae, you can see that they may just have, one haploid nucleus, or they can actually, so here's one nucleus, or they can have multiple nuclei in them, or be heterokaryotic, right? Most of them are going to have two nuclei in them. Each of these filaments is separated into cells by cross walls called septa. Okay, so here's your septa, and they allow for communication. So there's gaps in the septa called pores, and they enable materials to flow between the compartments from cell to cell. So nutrients can move rapidly through the pores. So, all the way out here, these cells can absorb nutrients, and they can move through the pores to nourish the entire mushroom. So here we see both the reproductive structure and the mycelium. So the reproductive structures, which we'll talk about later on, composed of hyphae. Okay, it can be very dense here in the kind of stalk area. Here's a nice picture of the different compartments, okay, the different cells that are separated by septa and the pores in there. And here again, we're talking about multinucleated cells. Inside of the hyphae, you can see there's multiple nuclei in each of those cells. These ones specifically, these conocytic hyphae, have hundreds of thousands of nuclei through their mycelium, so pretty cool. It's really important that these mycelia have large surface area because they are doing a heck of a lot of absorbing. Okay, so the more surface area you have, the more you can absorb. So lots of branching networks of very thin hyphae lead to a very high surface area to volume ratio. Great for water absorption, very efficient at nutrient absorption. The downside here is that these are very prone to drying out. So they're very, fungi, you always see them generally in very moist places. When it gets too hot and dry, we start to lose our fungi. So dark, moist places are the best for fungi. Okay. These hyphae produce, in many cases, we're going to see reproductive spores that can then, allow the fungi to spread and reproduce throughout the area. Spores can go into sort of a dormant, dry period and then germinate when the conditions are improving, which is why you can see mushrooms going away and coming back when the conditions improve. Okay. So specifically, the mycelia are going to, be an adaptation here that supports external digestion, absorptive life cycle. We're also going to see that fungi produce thick, fleshy reproductive organs. And something else that's unique about fungi is they can produce both sexually and asexually. So many species do not even produce sexually. It's easier for them just to produce asexually, which we'll see. There are important morphological differences among the lineages and species that produce sexually. So they've adapted to that, and we're going to see those differences here in a second. So let's look at some of those differences. So specifically, if we're talking about sexually reproducing fungi, right, sexually reproducing fungi, and the example that is given here first are chytrids, okay, which are these here, and they produce swimming gametes and spores, these chytrid fungi. And really interesting that these gametes are actually motile. So we've got gametes, and they have flagella, which allows them to swim. So we've got swimming gametes and spores that can come together through fertilization. And this is for chytrid fungi, like I said, is the specific type here that we're talking about. Chytrids also, like we're going to see in a second, do an alternation of generations life cycle, which we may have seen, I believe, already in the last chapter. We talked about that a bit, but we will get to that. Zygomycetes, they reproduce with a distinctive spore-producing structure called a zygosporangium. And these are formed from the fusions of cells joined together, and the haploid hyphae form two individuals here. So when these two haploid hyphae from two individuals meet, they kind of branch out to each other. They touch and they reproduce that way. So again, two different examples here in sexually reproducing. The third one, the club fungi. So these are the ones you're most familiar with, right? They have that sort of dome on them, mushrooms. Basidia is the, name for them, or basidiomycetes, form basidia, specialized club-like cells that form at the ends of the hyphae. So there's these basidia, and each of these basidium is going to produce basidiospores that will then go on to reproduce. Okay. Ascomycetes, or sac fungi. Now, if you're also taking the lab, or if you've taken the lab course before, or if you will take the lab course, you'll examine these in the lab. If you're not taking the lab course, I'd encourage you to, spend some time in your book and look at the different pictures of these, and we'll go over them in the last couple of slides. So you get an idea of what these look like specifically, and if you wanted to go out and even in your yard try to find some, it's not a bad idea to do that either. These ascomycetes or sac fungi form asci, reproductive sac-like cells at the ends of the hyphae, so little sacs with these ascospores in them that then will go on to reproduce. So these are specialized cells that produce the spores. Okay? And like I said, this is examples of sexual reproduction. But many fungi have also adapted to do asexual reproduction as well. All right. For fungi that use asexual reproduction as well, or exclusively, they produce asexual spores called conidia. Now, these conidia can be dispersed by water, wind, and basically just need to find the right conditions, and they can start growing into new hyphae, exactly like the original parent. So, whereas here we need, spores of different gametes, spores and gametes to come together, in this case, we don't need that. Okay, so just the one singular conidia will grow into a fully developed fungi. And we see this in blue cheese, conidia producing in there, so pretty cool. Okay, so this variation in reproduction has served the needs of the fungi, depending on where they are. So fungi, like I've said, have evolved many different ways to reproduce. So swimming gametes and spores, the yolked hyphae in which nuclei form different individuals, okay, to form a zygosporangium inside a protective structure. We see that. Specialized spore producing cells like the basidia and the SI. So, we see these various different forms of reproduction, again, depending on the location and what is best for the species of fungi. So these hyphae really come into major play here with mating and reproduction. So in many fungal lineages, hyphae come in different mating types rather than morphologically distinct sexes, right? So we don't see fungi having, a male and a female sex. You might wonder where the gametes come from, where the spores come from. It's not in the same way as a male and a female. So hyphae of the same mating type will not combine during reproduction, okay? So in this way, the mating types function as the sexes. So instead of just having two distinct sexes, a single fungal species can have, tens of thousands of these mating types, okay? So rather than just getting, male and female coming together, we've got all of these different mating types, and we're seeing a whole lot of diversity here from this. So really, absolutely incredible. So I mentioned spores. The most fundamental reproductive cell in the fungi is the spore. All right? They are the dispersal stage in the fungal life cycle and are produced in both asexual and sexual reproduction, as we've talked about. If a spore falls on a food source and is able to germinate, a mycelium begins to form. Okay? As the fungus expands, hyphae grow in the direction in which the food is most abundant. Spore production allows starving mycelia to disperse offspring to new habitats where more food may be available. So this adaptation of the spore as the key reproductive cell is very important in the life cycle, which we'll see here, in the multiple ways in which these fungi can mate. Okay, so how does fertilization occur? So I mentioned chytrids as being a specific type of fungi. They're the only fungi that produce gametes, like we saw, and they're not different enough in size to be called egg and sperm. All right, but we're going to see that fertilization in other fungi is going to occur. It's going to occur in two distinct steps, separated by long distances and long time. So fusion of hyphae and fusion of the nuclei from the fused hyphae. Okay, and there's a couple of terms here I'm going to point out to you that you need to know in the life cycle. And those two terms are plasmogamy and karyogamy. So the process of sexual reproduction in fungi begins when the hyphae form two different mating types to form a hybrid cell. Okay. In plasmogamy, the cytoplasm from different cells fuse, and the nuclei actually remain um, uh, independent. Okay, so in this case, you have one cell that's got two nuclei. It's called a dikaryon, and we say that its ploidy is n plus n. So it's not haploid, it's not diploid, it's a whole nother thing. It's one cell hooked together with two nuclei. In the second step here, karyogamy, eventually this pair of nuclei are going to fuse to form a diploid zygote. Okay, the fused nucleus is then going to divide via meiosis, and then it's going to produce spores. So karyogamy, again, the fusion of the two nuclei. Okay, it's no longer n plus n, and it's now a true diploid cell. Okay. So many fungi, like I said, reproduce asexually. Okay. The spore-forming structures are produced by the haploid mycelium, and in this case, the offspring are genetically identical to the parent. Okay. Microscopic spores carried by wind is an adaptation to life on land that allowed for the vast dispersion of fungi and the ability to colonize new habitats. Okay, so here's a look specifically in this figure, showing the asexual and sexual reproduction side by side. So asexual here on the left, and sexual reproduction on the right. Okay, so if we look here and we focus on our let's see where we want to start here with this one. Let's start with our mycelium here N. And your mycelium is going to undergo mitosis to produce more of the same. Okay, on its end, it's going to produce spores, spore-producing structures, which will undergo mitosis and produce more spores to ultimately grow back into the mycelium. So that is the very simple asexual reproductive life cycle. As long as the conditions are right, these can very easily grow into the mycelium. All right. So in the case here of plasmogamy and sexual reproduction, we have fusion of cytoplasm from different individuals. Okay, and we end up with this heterokaryotic nucleus, this n plus n. Okay, so here's the two nuclei per cell. These are going to fuse the two from different individuals and produce a diploid zygote that will undergo meiosis. Okay, it undergoes meiosis here again, very similar to what's happening here, but the difference is that now look at our spores. Some are dark and some are light. We end up with, again, haploid spores, but the difference here being that these spores are not all the same. Okay, so interesting. Make sure you recognize karyogamy, plasmogamy, heterokaryotic, those terms, and how they kind of fit into this life cycle. Again, you'll be able to reference these on your homeworks, and you won't need to memorize it, but you should understand what is happening. Another kind of key characteristic of the fungi is their ability to participate in symbiosis, or living closely with another organism. So the first plants we see in the fossil record showed this symbiosis, okay, with fungi. And it was probably very crucial to the early evolution of land plants. So about 90% of land plants today live at least in some physical contact with fungi and have a symbiotic mutualistic relationship. Again, in terms of real review here of ecology, mutualism, so symbiosis is any time we've got two species living together. Mutualism is a mutual benefit to both species. So in the case of the mycorrhizae fungi, they're both benefiting. Okay, we see this in a lot of animals and plants and all kinds of different organisms. Parasitic relationship, we've got a parasite-host relationship where one species benefits at the expense of another. And in a commensal relationship, one species benefits while the other is apparently unaffected by the relationship. But we may just not have discovered that benefit. And I'm assuming you've seen this before at some point in your education. Right. Another really great, kind of classic symbiotic relationship we see is between a species of Ascomycete and either a cyanobacterium and an algae. So we've got here lichens. Okay, so these lichens, I'm sure you've seen these on trees and things like that. and very easy to find. And like I said, if we were doing the lab, you would uh, be looking at these, but you could probably find lichen between a photosynthesizer, an algae, and most of the times a tree, and you can find these pretty easily. All right, so here's the key lineages of fungi that want to recognize, and I have crossed out here the ones you won't see on your exam, but we've talked about the zygomycetes already. This is what they look like. They've got those asexual sporangia, and these are basically your food molds. Okay, so these are primarily soil dwelling. They live on plant debris. Some are parasitic. They affect insects, spiders. Um, and the hyphae of different mating individuals will fuse during sexual reproduction in the case of the zygomycetes. We did see that earlier. Really common here, you'll see these as being common black bread mold as a pest and things like that. So really want to recognize that one. Okay. Uh, Basidiomycota are your club fungi. These are your mushrooms. Okay, you're really kind of common. Your puffballs, stickcorns, ball eats, brackets. Okay, your club fungi, Basidiomycota, very important. These actually can completely digest wood. It's kind of interesting, that lignin, which makes them important in the carbon cycle. Some are parasitic, some are EMF, some produce basidia during during sexual reproduction. They have heterokaryotic mycelia. These are again really important in forestry as they do help recycle that carbon and improve the productivity of forests. Ascomycota are your sac fungi. There they are. So these are found pretty much everywhere from what I remember. They use every possible absorptive life cycle that you can think of. They grow in symbiotic relationships with the lichens like we saw. They protect algae, cyanobacteria from desiccation. They produce spores called conidia like we've seen. And they're really distinguished by this kind of cup structure, the sac fungi, where that name comes from. Okay, and these guys are the source of penicillin. Okay, so you can thank them if you've ever taken penicillin. You also get truffles and things like that from the sac fungi. All right, so that will end chapter 29 lecture. Thank you for your attention.